Hello everyone. My name is Dr. Alan Carlson. I'm a research fellow at the Hearing Research Group at the University of Sheffield. And today I'm going to talk to you about how I'm spending the next 5 years of my life investigating how the brain talks to the ear. That's it, I'm finished now. Right, so just briefly I'm going to go over how hearing works, just because I forget that not everyone's an auditory researcher for some reason. So, swap sides. So, as you imagine, this is the ear. That'll be all familiar. So, sound hits the ear, moves into the ear canal, hits the eardrum. That then vibrates, and through these three little bones, one of which is the smallest bone in the human body, the stapes, this one, the sound information is then concentrated into what my wife calls the snail in your ear that talks to you, which is the cochlea. So the sound is basically, concentrated in this big drum, this tiny window. And if you were to crack open the snail in your ear that talks to you, you'd see something like this. Less blue, that's for artistic license. And if you look at this kind of spiral, you see the actual hairs. So you may have heard about the hairs in your ear that actually detect sound. And this is what people are talking about. So if you imagine the stapes is like here-ish, which is about here-ish, the sound comes in, it moves all the way up the spiral. So this spiral is organized tonotopically, and that's just a fancy word to mean high frequencies down here, low frequencies at the top. So if you were to hear something like a rock concert noise, for example, that's a really complicated wave it's doing to your ear. And what this is doing is basically breaking it down. So if you imagine like the singer has a really high-pitched voice, for whatever reason. The singer's voice would be down here someplace, and the kick bass drum would be all the way up here. So it's taking the noise and it's breaking it down into its components in a way that our brain can understand. Because if you imagine all this information going through one cell, it would just be a complete bottleneck. We wouldn't get anything out of it. So like I said, these little hairs, swap sides again. They're very, very small. The scale bars here about 2 micrometers or 2/1000ths of a centimeter times 2. Yeah. And very, very dinky. These deflect forwards and backwards in response to the vibration of sound. And that passes information onto nerves that go into your brain. And that's basically how all of sound transduction works. It took us a lot longer to find out than I was to explain. But this bit is quite intuitive. I'm sorry, it's kind of so sorry. This bit's quite intuitive in that you imagine in the big snails down here, this is in the snail, sound hits the little hairs. And then that information is being broken down and it moves from the cell into the brainstem here and then into the higher areas of the cortex. So imagine this is where you live. This is the autonomic bit that you never really speak to but does things for you in the background. And this is the thing that detects noise. So we understand relatively well how this bit works, how information flows to your brain, we do know pretty well. What we don't know is the little green bit here. So that is why I investigate. Basically, the brain talks back to the ear, and we're not quite sure how it works. So the reason this is there is to give your ears and your brain the ability to kind of focus on specific noises. So the ability to pick things out of your environment, amplify them up so that you're more aware of them. So it doesn't do this directly, as you'll notice the little green wire on my little cute cartoon here doesn't actually go to the sound detecting cell itself. It actually goes to the wire. And that is how this works. That's the basis of this feedback, because it's always running to the wires, not the actual cells. So the reason we know vaguely what this does is from very, very old school experiments. And like all old school experiments, is basically we cut something with a scalpel. How does the animal react? Not great, as you'd imagine. But for the most part, what is lost when you cut these little green fibers is that you're more sensitive to noise exposure. So if you listen to loud sounds in the same way that you would a rock concert causes damage, that's a lot more damaging if these fibers aren't there. Your ability to localize sounds in a noisy environment is completely disrupted and things like that. But it's very cloudy because, as you can imagine, taking a scalpel to a brainstem isn't exactly that precise. So because of this and because of how we investigate hearing to begin with, this feedback is probably the most elusive aspect of hearing that we still have. The humanity just does not understand just yet. So the way I'm going to break it down, and the way that will not be written anywhere else but here, is that this is broken up, this feedback, the green cells in the previous one, is broken into two parts. And I've nicknamed them the equalizer and the volume dial, because that is basically how they work. So, can you imagine, like, it's basically the exactly the same as an old school audio equalizer. So, the left is your bass frequencies, your right is your high frequencies. And what these cells are doing is they're basically amplifying one over the other. So it's letting you pick out specific frequencies in noisy environments. So like I said, that's by frequency or by pitch. So in the same way I said before, the singer at a rock concert has a high-pitched voice. By focusing on that, your brain lets you actively hear that more clearly. It amplifies that sound relative to everything else. So that's the first aspect of the system. The second one is the volume dial that I've nicknamed. But it's a volume dial, right? It's a volume dial that can only turn down. So this is basically a global, that's loud, shut this up type cell. So you can imagine that's how these two systems are opposing each other. So how do you train this? So if you imagine, if you hear something quite faint and you want to amplify it, the equalizer kicks in, amplifies that right up relative to everything else, so you hear it clearly. If that suddenly gets really, really loud to the point it's going to damage your hearing, the volume dial then kicks in to dampen everything down and protect your hearing so that you're not deaf for the rest of your life after hearing whatever high-pitched singing you just were listening to. So these are both fighting each other to make everything basically just as sensitive as possible, as effective as possible, but also protect everything in the long run. And both these systems reside very close to each other in the same nucleus, or just basically a big lump of cells in the brainstem. So the first one is the equalizer. I've put the official name up there. I don't expect you to remember it. It's just intrinsic lateral Abekopfer neurons. I nicknamed them ILOCs, or the equalizers. So they sit within the core of this nucleus. It's like an S-shaped nucleus in the brainstem, but does have quite a distinctive S. And they're arranged in the same way that the ear is, the snail, from high to low frequencies. And this is what it actually looks like. This is an image I took. I just labeled them to make them very fluorescent and not at all natural. And this is what you see. So what you can see here is that there's lots of them. And there's lots of them because there's a lot of frequencies to cover. Humans have quite a wide frequency range that they can hear audiences. So they're down from, I believe, like 200 hertz all the way up to 20,000. There's a lot to cover. So each of these cells is responsible for one small patch. And together, they give you a very good resolution of the whole of your hearing frequencies. So if you imagine this is your kick drum, this is your singer, and if you want to hear the singer better, these cells kick in to turn that all the way up to let you hear them relative to everything else. So they're, like I said, they make hearing more sensitive to certain sounds. There's two ways in which this works. The first is by reflex. So even without thinking about anything, if a noise comes into your ear, your brainstem is working to make that as clear as possible for you to perceive. That's just an unconscious part. There's nothing you can do about it. But there are also inputs from the higher areas where you live, like I said before, the cortex. So if you consciously think about something, you can hear that better than if you were not paying attention whatsoever, which is kind of intuitive. And like I said, the reason I get from the names, because it works similar to an audio equalizer. But by default, and the reason this green bar has been there this whole time from talking, is because these can only amplify sounds. The orange marker I've labeled here in this funky little diagram is excitatory. It means it's switching things on. So it's purely activating those little nerves. It's setting their resting state almost. But it's not quite as simple as that, like everything else in my science, in that based on the history that you've had, they can kind of switch gears. So let's say you had some kind of noising source. You had a really, really loud sound at some point in your life, around 5 kilohertz. So you kind of mid to high frequencies. The cells in this feedback system that are responsible for those frequencies will have switched as always a protective mode. So they change the nature of the feedback to make it protective. So instead of switching things on, they're now switching them back off again. And the reason they're doing this is to protect the very sensitive wires that carry all that information from the ear back to the brain. So they've got these kind of two modes. So if you imagine it in terms of an equalizer, I'm going to switch back to the first guitars, forgive me. So there's the standard. So obviously 0 dB and up, they're amplifying noises. Hopefully familiar with equalizers. And if you were to be, for example, in this case, exposed to a noise in all frequencies, the way in which these notes work will then shift down. So they're now able to not only amplify sounds, but they're now able to shut them back up. And it's basically, it's hearing working, like I said before, in a balance between keeping you sensitive versus keeping you not deaf. So this is the bit that I really hope works, because this is going to be quite a bit audacity, and I'm not great at that. So, for example, in terms of when I was talking about the unconscious part, so you hear a nice piece of music. For this example, it's my favorite song. Whenever you're thinking about it, you're hearing this. So your hearing's kind of in that autonomic thing I was talking about before, the reflex. You're listening to that, you're probably subconsciously focusing on the singer, because we as humans, we hear people speak, want to know what they're saying. So you probably got a few words out of that, and you were picking her voice out from a very complicated pattern of sounds. I mean, there was drums, towards the end there was violins, some of this. It was a very busy frequency, and you were able to pick that out. But let's say, for example, and I really hope this works. Let's say, for example, your niece, for whatever reason, is playing the bass guitar of that song, and you really want to listen to your niece play. In the same way that equalizer works, you're basically filtering out all the higher frequencies. So this is going to sound a bit weird, because I basically just turned up the low frequencies and turned down the high frequencies. That's basically how it works. So your S-shaped diagram sorts out. So in that example, and this is probably what the activity was looking like in your brain stems just then. The neurons at this side, the lower frequency side, would have been kicking in. They would have been trying to pick out the thing I was speaking about, trying to hear this phantom bass guitar and that very bassy mess that I made. Because there's conscious input. You're able to selectively amplify sounds. I might skip through this, that doesn't sound too great. But basically it's saying so high frequencies.
My intention is to fade, and I will
You didn't hit the violins. I probably hit the violins. But they were higher frequencies, basically the same thing. On stuff. Now, where did I get? So that was the one aspect. And that was, as far as humans are considered, as the better understood aspect of the feedback that I'm talking to. Just because these ones, the shells, or the volume dials, were discovered a lot later. We've only known these things have existed for like the last 10 years, maybe? Just because the technology didn't actually allow us to even know they were there. So this is a relatively new finding as far as science is concerned. So in terms of the ball of cells, or the nucleus I was talking about, these reside around the outside, which is why they got the nickname shell. But for the sake of being a bit more intuitive, I'll call them the volume dial. There's not many of them, so this is kind of reflecting their function. This kind of reflects on their function in that the other ones were kind of like a big piano, right? So you want to hear the lower frequencies, the lower ones play, amplify sound. These ones are more of a global, that's too loud, this is going to damage my hearing, but then kick in. In that they extend out the snail and cover a very wide area of it with this kind of dampening projection. They're kind of making everything quieter to protect you. So they don't need that frequency selectivity, because if you hear something too loud, you don't care if you want to focus on this or that. You just want to protect your own hearing. So because they're so few and because they were so hard to pin down in the first place, we're not really sure how they work, like at all. Nobody has ever recorded from any of these. They were only identified recently, and there's about, I want to say like 10 per side, but not very many at all. We'll think about how many cells the human body has. So in this, similar to this kind of diagram that produced on the left, because this is how we know their wiring looks, we can kind of make some assumptions as to how they work based on this. So based on the fact that they're dampening sounds down. But everything else is just guesswork. Nobody knows. This is another image I took. I got a bit more creative with the colors this time. But the shell cells, so this is your S-shaped nucleus here. These are all the equalizer cells nicely lined up. This would be your low frequencies, up here would be your high. And in all of these cells, in all these hundreds of the equalizer neurons, there is one of these shell neurons, one of these protector neurons. Which is why I've just been so hard to study, because like, I used to call this isolate, that's cozy. So that is the reality of what it actually looks like. So that's how we think they work. How we think they work is that don't worry too much about the beta this one. But basically, the equalizer is always set above 0 decibels. It's always amplifying everything. That's its basal level. So that when this reflex pathway kicks in and you hear something, it can make something louder or quieter on the fly. It's not having to switch into this protected mode all the time. So this, because it has this kind of constant input to the stellar area, it gives the brain like a really fine control over all the frequencies. So a good example for a place like this would be you talk to your friend in a bar. Your friend has a relatively high-pitched voice, and at the bar, there's lots of men talking, so everything's relatively low frequencies, allowing you to pick them out selectively based on the frequency. That's the kind of real-world application of this. So that's the kind of the specific aspect. When a loud voice is encountered, so a big train goes past and it's over 100 decibels, for example, that's when the volume dial kicks in and says, right, enough of that. This isn't about survival anymore. I want to damp things down. So it keeps things sensitive, detects from noise, and it lets us focus on things based on frequencies. But the whole reason I got into this research in the first place is because there are things that happen with this feedback when it comes to the ear, when it comes to the snare that is in the ear, that almost suggests this system is some way involved in aging, in that it almost switches back to what it's like before you can even hear it. But imagine pre-hearing in the womb, that kind of wiring comes back as you get old. The same pattern. Nobody in the world has any idea why. And the whole point of this research was, I was thinking, if the activity in the equalizer, for example, looks one way when we're in the womb and developing, and looks another way when we're mature, as I am speaking into you all now, does that go back as well? Do these neurons in themselves almost become immature as we get too old? Was the whole kind of reasons. That was what this diagram's trying to show. Don't worry about it too much. The point was, before we can hear in the room, they fire really spontaneously. Just once every now and again they fire. It's not very frequent. This is the kind of resting activity I was trying to show you. So the equalizer is constantly above 0. This input's constantly there. The question mark is the whole point of my next 5 years, basically. That was what I wanted to investigate. So the whole point is, and a biggie, especially around trains, would be noise-induced hearing loss. So it's very common. You all probably know someone that suffers with noise-induced hearing loss. I'd be surprised if you don't. So for example, here's me with my cause of noise-induced hearing loss, and we're both sat on a bike. So it's very, very frequent. I'll probably suffer from it because I have a motorcycle, for example. As you'll probably be aware, because you'll do those silly little YouTube tests where it's like, how high does your frequency go? And you feel terrible about yourself. They usually start, the hearing loss usually starts at higher frequencies, and it's perfectly normal for us to lose those high frequencies as we get older. It's why teenage alarms are a thing, and not just something that adults have to put up with. And that happens as these wires that this feedback's going to start to die away. So for every cell in your ear that is actually detecting sound, there are 20 to 30 wires coming away from that to give to your brain. And you won't notice the first 5, you may not even notice the first 10, but once they're gone, they're gone. And once they're gone, the cell itself dies. You lose that frequency forever. And it sounds so scary with that. Sorry, that sounded awful. But the reason that you can go quite a long time without noticing this high frequency loss is because we can deal with it really, really well. Our brains are remarkably plastic. You can pull up with incredible insults to your body and just keep going. It's just the way humans seem to be. So you can you can master somatically well right up until it starts to hit severely these communication frequencies. So this is what a normal human audiogram looks like. So literally you just tell the person, when can you hear a noise, skip the bunny, hear the button. And you can create this graph. So everything above this line, you can't, no? Yes. Everything above this line you can hear, everything below this line you can't. So if you imagine as you age, this line of the right ear is just gradually forcing yourself over here, and this is the part that's mainly used for conversational speech. So right up until it takes a big chunk out of this, you probably won't notice any high frequency hearing loss. It'll just be on the silly YouTube videos linked to the bad right. So, but there will be a point where you start to notice it, and by that point you'll already be pretty strongly into the conversational part. So that's why this is so important, is because if we can find a way to stop this from happening, then everyone can live a lot older and hear a lot better. It's such a good timing, because we know it's already doing this. We know that this feedback is already protecting against noise-induced hearing loss. That's already well known. So if we can kind of understand it better, if we can understand what makes it tick, how to activate it, how to switch it off, if we can manipulate that finally, maybe we can use it to prevent noise-induced hearing loss. The noise, kind of. Part 2, this is a bit more exploratory, but very for anyway. Tinnitus is probably something you're all again familiar with. That high-pitched ringing you might hear after a rock concert, or just all the time if you're unfortunate if you has it. And it comes in 2 flavors. So there's subjective and objective. So subjective is the one I'm going to focus on. Objective just means there is an actual noise. You are hearing something. And that's arising from the middle or NOA. But the subjective tinnitus is basically when those wires that we're talking about, the ones in blue on the bottom left, when they start to die away, the brain suddenly lost the signal it was getting before, because they're not silent in silence. So if you think about, if you sat in a perfectly silent room, all the cells dragging this information away from your cochlea they're still firing, they're still sending signals hundreds of times a second, and that is the code for silence. And it's only when you lose that code that the silence is something you just can't have anymore, because you're constantly trying to amplify the damaged wiring to bring hearing back. So that's where the hybrid ringing is. It's an overactive compensation almost. It's a good thing. It's just gone too far. So, like I said before, if you could manipulate this resting activity, if you could bring back the sound of silence using these neurons that I'm so obsessed with, then maybe you could trick the brain to stop trying to amplify it, and maybe you could switch off tinnitus. This one's a bit more exploratory, real activity. So what I'm doing, as I said, the way this was studied before, and the way it continues to be studied now, is these connections between the brainstem here and the cells that detect sound have been cut. So that means if you're trying to investigate these cells, you will have no idea what my neurons are doing, because they completely disconnect. So studying these have always been kind of a byproduct in the past. So what I want to do is, instead of looking over here and these are the byproducts, I want to look solely at my neurons of interest, the feedback, the equalizer and the volume dial, to understand what they're doing during aging, during deafness, and even just when everything's hunky-dory, what are these cells actually doing? But yeah, as I said, looking at the volume dial neurons, like I said, have not been studied in detail all up until now. So that's absolutely something I want to do, because it would be very interesting. And hopefully, by the end, I'll be able to kind of build a model of how this is all playing together, how this all works. So I use electrophysiology, which is basically just a long word for stab cells or a little bit of glass to figure out exactly what they're doing. This is kind of what it looks like. So at the top left is an image I took. It's a slice of brain. You focus in on the clump of cells, which is that little light bit you can see. And then you can record what these equalizers and these volume dial nodes are doing. And so far, I've already kind of bubbly things up by disproving the whole constant firing thing. I hope the lad that found that aren't too upset with me. So I found that it's not really a thing. The equalizer isn't set above 10. It's always set at 0, and I'm not quite sure how they found what they found, but I'm not going to argue too much. So I didn't see this. I did see, however, that as as you mature, so as you gain the abilities here, and as you age, they get stronger and stronger. So they're turning up the amplifier higher and higher, almost to compensate as your hearing starts to fail, and to give you the ability to hear when things start to go a little bit wrong. And the kind of the rewiring, the all immature versus mature of the womb versus adult thing seems to be completely independent, which is annoying because that's what I was supposed to spend the next 5 years doing, but I've got lots of plans to do it time. So basically, unless science goes most of the time, all of my hypotheses turn out to be accurate. I've still found things, they're still interesting, but it's not what I thought was going to happen whatsoever. So I'll do my best, but it's still all positive data. It's just in the way which it always is. We're building pictures with imperfect information. We're telling about the gospel, but they really are. So it comes to my conclusion here. So as I said before, there are two distinct types of feedback that are coming from your brain to your ear, and they're constantly fighting each other to protect your ear and make it more sensitive. They're both elusive just because of the fact we didn't know they were there. We have no idea what they did. Whenever you start to investigate this, you always take it out of the mouse, out of the gerbil, and my neurons feel left behind. So you're not going to have any idea what they're doing, irregardless. This feedback is not responsible for hearing loss tinnitus, but because it assists with both, if we can manipulate it, it may open up new treatments for preventing both. So it won't bring anything back to life by any means. But, for example, if you knew you were going into a loud rock concert on day 2, and this is something we have that our biology doesn't, it's foresight. So you knew you were going into a rock concert. If you could turn the volume dial down a bunch and dampen down your hearing to protect it, would you do it? Would you manipulate your own biology even protecting your own ear? So these are the kind of options that may or may not open up in the future. I'm not sure. So at the end of my contract, in about 4 years' time, I should hopefully, if I do a good one, be able to tell you a lot more about how these work, because things are so mysterious and I am working with imperfect information, but I hope I'll at least give you an idea of how the two systems play with each other. So while I am technically a lab of one, talking in the middle, I'm not by myself. The hearing group in Sheffield is huge by international comparison. And we're only getting bigger and we're only getting stronger. So this is the hearing group with some of my little family of nerds. And yeah, it's a very good place to work, and I hope to stay in science for as long as I can. So yeah, I welcome any questions. Oh, that's it for me.